Hello, everybody. My name is Dr. Jennifer Stone. I'm a Senior Manager of Product Marketing in the Oncology Business Unit at Illumina. And today it's my great pleasure to share with you some information regarding the development of technologies to really transform oncology with genomics. So to start us off, just a little bit of review information for those of you familiar with this space. The war on cancer was declared almost 40 years ago now, over 40 years ago now. And as you can see from this chart here, despite billions of dollars and tons of research into this disease, the needle really hasn't moved too much in terms of overall mortality from cancer. You can see in the red line across the years here, it's stayed pretty consistent while there's been great strides in, for example, diseases of the heart, decreases there and with more scientific discovery. And so it really begs the question, what does the world, what does the community need to do to really understand cancer and be able to fight cancer more intelligently? It's a huge world health problem. It doesn't just affect the rich or the poor. It's something that we all have to deal with around the world with high incidence rates in Western Europe and North America, Australia, but certainly a situation that all of global health organizations need to deal with on a daily basis, with lung cancer being the highest, most prevalent cancer, and breast cancer a close second after that. But incidence is only one aspect of this equation here. Many people get sick. It's really a question of, can you effectively treat the disease, and can you control the disease such that people can live longer, healthier lives? And so on some level, perhaps the most important statistic here is going back to the mortality figures. If we look at breast, prostate cancer, there's high incidence, but actual mortality from those cancers is on the low side. Compare that to something like lung, where the incidence is extremely high and mortality from lung cancer is also extremely high. And so it begs the question, what can we do better to really move the needle on this disease? I put up on this slide a couple of recent figures, one from the US, one from the UK. About 10% of cancer in the US is diagnosed at stage 4, and about 50% in a recent report from CRUK show that cancer is being diagnosed at later stages. And this is an important consideration when we think about the options available for patients, as well as what we can do, again, to really change the paradigm and influence that mortality statistic that we looked at on the first slide. So the reality of cancer treatment, and this is an oversimplification, so I apologize to those of you who know this in intimate detail, but the reality is surgery is the only proven and established cure for cancer. At the end of the day, if you can surgically remove the cancer in its entirety, the patient is free and clear and can go on to live a healthy life. For those patients that are not detected until later stage or have unresectable tumors, really chemotherapy, some sort of systemic therapy or personalized medicine are two of the only available options to those patients. And this is an important aspect because a lot of the conversation that we talk about when we think about genomics and oncology is really focused at the use of genomic information in either diagnostic, companion diagnostic, or prognostic situations, so the patient is already diagnosed. What I'd like to share with you today is actually a vision that's much greater than that for genomics in oncology, a vision that includes understanding the role of germline variation in oncology, using our technologies to potentially crack open healthy screening and push back the timeline and the age and the stage at which cancers can be detected, thereby providing patients with a broader array of treatment options. To use our technology to monitor treatment progress and treatment effectiveness in real time. And to use our technology for more of a recurrence monitoring, a recurrence screening type paradigm, very similar to the healthy screening type concept. To this end, Illumina is working on 3 strategic pillars in the oncologist space. The first one is focused on germline variation, which has a critical role certainly in germline risk estimates, but is also increasingly important in diagnostic and companion diagnostic type scenarios. We're also looking at molecular monitoring as a critical area of product development, and as you can see here, molecular monitoring can potentially bring benefit down the road across at least 4 of these 5 stages on sort of a clinical care cycle for oncology. Everything from companion diagnostics, liquid biopsies, for example, monitoring treatment progress, again, recurrence monitoring, and then eventually someday, perhaps in the use of healthy screening. And underlying it all is a fundamental understanding of tumor biology, not only understanding specific mutations in the DNA, but how those translate into expression signatures, how the methylome looks. There's a huge complexity of cancer biology that is still being unraveled, and we'll talk through some of those in the coming slides. So starting first with germline variants in cancer, it's pretty well established, an estimate at least, that about 5 to 10% of cancers have a hereditary component. It's also recently been established that knowing one's germline status, for example, their BRCA1 and 2 status or their PTEN status, can be an important biomarker for various pharmaceutical interventions, for example, PARP inhibitors. To that end, Illumina has developed over the past couple of years a targeted panel called the TrueSight Cancer Panel. This has been designed in collaboration with Professor Nazneen Rahman at the Institute of Cancer Research in London. And the whole goal here is to design a reagent that allows for large-scale testing, large-scale sequencing of huge numbers of people so that we can really establish the statistical power to understand the role that variants in germline DNA are playing in the individual's cancers. There's still a huge amount of research to be done here, but some of the critical sort of usual suspect genes are of course included in this panel. So whether we're looking for colon cancer, breast cancer, kidney cancer, et cetera, melanoma, retinoblastoma, the usual suspects are all included here, and they're being used today in the research laboratory of Dr. Nazneen Rahman, among other leaders in this field, to really tease apart what's the role of germline variation in cancer. As I mentioned, also the role of germline variation is emerging as an important biomarker in the pharmacology space as well. And right here, I just have a few screenshots from recent papers that have been published that are exploring that fact and really building the body of evidence to support the testing of germline variation as a routine component of any cancer patient's workup. So moving on to tumor biology and tumor profiling. Here I have a figure that is referring to the great work that's been done by numerous organizations around the world, the Cancer Genome Atlas effort, or the TCGA program, as well as the International Cancer Genomics Consortium, the ICGC. Both of these efforts are huge international working groups that are looking to explore and really map out in detail the biology of what it means to be a tumor, what it means to be a cancer cell. And while there's many people involved here, and you can see, I think that's indicative of the geographic spread represented in the figure here, it's too much to go through at this point in time, but suffice it to say, some of the important findings that come out of this work really highlight not only the differences between cancer and the differences between cancers of different tissue types, but the similarities as well. And so we're moving to an era where instead of being solely defined on tissue of origin, cancers are certainly defined on that category as well, but also defined on their molecular makeup, both DNA variation, expression patterns, epigenetic patterns, etc. And all of this information allows researchers to really understand what are the strengths and weaknesses of a cancer cell or a tumor mass, and how can they be exploited to treat patients better. This particular analysis that I'm highlighting here was published last year. This is a paper that came out of the TCGA pan-cancer analysis effort. It was really a tour de force, and the first time that so many different tumor samples were looked at en masse across so many different tumor types. Historically, these sorts of analyses were done as a single tumor type in sort of an isolated and relatively small scale type of experimental setting. In this particular example, they looked at 12 different tumor types from the TCGA project, and you can see them listed there along the top, and looked at they performed an analysis on whole exome sequencing from these cancer samples and looked at numerous different factors about those samples, looking at differences in mutation rates, looking at the mutational spectrum and the mutational signatures of those cancers. How are they similar? How are they different? And as we add this additional layer of molecular complexity to our understanding of tumor biology, we can then again use this in more intelligent drug design, more intelligent therapeutic regimen design as well. Now, the TCGA project has delivered a huge wealth of information that's being used by researchers around the world, both within the TCGA and ICGC projects, as well as numerous other people around the world that are mining that information. But it begs the question, how much is enough? And have we really discovered all that there is to know about cancer cells, and can we just move forward into other lines of inquiry? And I put this slide in here to highlight the fact that there's still a huge amount to discover. This is a paper that was published earlier this year, Lawrence et al. In Nature, and they asked the question, of those data sets that are available for thousands of different cancers across dozens of different tumor types currently available out of TCGA data, what additional information is being identified with each incremental sample that's analyzed? And so as we look at the graph here, going along the x-axis, starting at 0 and going up to close to 5,000 different tumors, we see that the number of new variants, new genes being identified at a mutation frequency, for example, at 20%, the red line, it's kind of leveling out. And so that's indicative of the fact that we've really found, as a community, the genes that are mutated most often in cancers. 20% and above, we've identified those genes. If we change and look at genes that are mutated less frequently, so let's say 3 to 5%, looking at the light blue line, you'll see that that's increasing and no sign of leveling off. And this is suggestive of a huge amount of additional discoveries that need to be made that are in the lower frequency range, meaning those genes that are not mutated in a high fraction of patients or a high fraction of tumors, but they are significant in a non-trivial fraction of the overall cancer population. And so the conclusions of this paper were, while highly mutated genes that are mutated at high frequency, have for the most part been found, based on this analysis, intermediate frequency down to about 2%, for example, will require huge numbers of samples to complete this catalog, to really understand what the full spectrum of mutations are within what we call quote-unquote cancer, which is, as we all know, an amalgamation of many, many diverse different diseases. Their estimation would be that you need about 2,000 tumors from each of about 50 different tumor types to really map this out down to about 2% frequency. And this corresponds to about a total of 100,000 different tumors. So as these projects continue on, they'll clearly be making more and more discoveries and adding to this body of work. Now, I should point out that this is just looking at DNA variation. It's not even touching the role of RNA or epigenetics, and those are additional very important lines of inquiry that are going on by TCGA, ICGC, as well as many other groups around the world. I do want to highlight, though, that one of the one of the comments that I often get, and that many of us often get, is, well, you know, what's the importance of a gene that's mutated in 2% of patients? What role is that really going to do for anybody? But I do want to highlight that crizotinib, which is a really successful personalized therapy, is targeting ALK translocations, which are only present in about 3 to 7% of lung cancer patients. So, you know, not high on the mutation scale, those ALK translocations, but a clear success story for the development of personalized therapies, and exemplified by the story that we've seen with crizotinib. So I would argue that going down to 2% and understanding the genes that are mutated at these intermediate frequencies is an important endeavor for the world community to continue. So in summary, for the discovery work on tumor biology, there's been over 120 significant mutated genes that have been discovered by the TCGA group. And there's been over 550 genes and 120 different fusion genes identified in the COSMIC database. So while we've been able to take the full catalog of the human exome down to on the order of hundreds of genes that we're interested in, that's still a significant number of genes that need to be followed up and investigated in more of a clinical research context. So how do we take what's emerging in terms of the complexity of cancer biology and try to push that along the product development cycle to really develop next generation clinical products that bring these discoveries to bear in personalized medicine? This is a process that's going to be ongoing for many years. And something that we are trying to support and have tried to coin the term or use the term that's been coined by other people of theranostics to describe the work that we're doing here. And it really starts with one of the challenges that pathologists and oncologists are facing today. And today they're facing a challenge, just a workflow challenge, really, in getting patients access to personalized therapeutics. For example, in this slide, we've got a hypothetical lung cancer biopsy. Let's say it gets tested for EGFR. It's either wild type or it's not. If it's wild type, we then maybe test it for ALK translocations. It's either wild type or it's not. And so you can see there's this sequential testing that happens to a potential patient sample. And this iterative testing increases demands for tissue that's always in scarce supply, certainly increases the time to action, and time is precious for these patients, and costs, overhead, labor to support all this sequential testing. The reason for this paradigm is that the majority of these companion diagnostic tests, in fact, all of these companion diagnostic tests to date, have been developed starting with the drug first. So there's a drug that looks promising. The pharmaceutical companies and clinical researchers that are supporting bringing this through clinical trials look for a biomarker that's suggestive of action in a subset of patients, and then that biomarker becomes the companion diagnostic. That's the paradigm for how it works today. The challenge is that thus far in the United States, there's only been 12 companion diagnostic tests approved for personalized therapies in cancer. But the pipeline is huge. And so we're looking at somewhere over 500, depending on who you ask, personalized therapeutics in development today. And so if we take the complexity of the pharmacopoeia that's coming down the pipe, in addition to the complexity of cancer that's emerged from the recent studies that we just walked through, you can see sort of the factorial combination of all those different variants plus drug combinations, et cetera, becomes an intractable problem, again, when you're dealing with limited time, money, and available tissue. Certainly not extensible, not achievable, not explorable given the current paradigm of testing. So one of the things that Illumina is working to try to support is changing that paradigm to start with the patient. Patient-centered oncology therapeutics, where the patient starts at the center. The next step is then understand the biology of that patient's tumor, really understand the mutations that that tumor is carrying, and then use that information for informed drug selection. And use that to really pair the patient to the right drug at the right time, saving valuable time, money, and labor for the patient and the doctor. So changing the paradigm, instead of having this sequential one-off testing, the future that we're trying to develop tools for looks something similar to this, where there's a cancer biopsy. This Universal Oncology Test is then applied to that cancer biopsy, and there's either an actionable variant with an approved informed therapy available, or if there's no actionable variant detected, potentially looking at additional variants and then enrolling that patient into a clinical trial if available. And so it really cuts down on the waste right now, quite frankly, of tissue, time, and money to support patient testing. Now, Illumina is certainly not the experts in cancer biology. We're our technology company for historically, and so we've engaged the expertise of a number of different cancer centers around the United States. We've formed the Actionable Genome Consortium, which was announced a few weeks ago, including Dana-Farber, Fred Hutch, MD Anderson, and Sloan Kettering. All four of these institutions are providing insight into what is truly actionable, and how do we define a set of genes and a set of criteria that can be standardized and shared with the broader oncology and pathology communities. But certainly the pharmacopoeia is the other side of this equation, and essential to making this entire strategy work is to pair both sides of this equation together. And so on the pharma side, we've announced 3 partnerships, one with AstraZeneca, one with Janssen, and one with Sanofi, to really work to develop a universal test that can be used in clinical trials and put on proper paths towards regulatory approval. The goal here is to develop an ecosystem where the universal test and the patient are at the center. And important players in this entire exercise clearly are the pharmas, as we've already discussed, and the cancer centers, but engaging with the FDA early and often, as well as members of the extended Cancer Center Network and Clinical Trial Network across the United States, bringing in biotechs as well as the CROs that perform much of this clinical trial work, with the goal of standardizing, something that's lacking in the clinical development of NGS today, streamlining such that this massive pipeline of new potential therapeutics has a very clear path to companion diagnostics, and the introduction of new biomarkers can be done in a much more streamlined and efficient way. Decentralization to allow genomic testing via standardization and the streamlined aspects of this program, decentralizing it closer to where patients are actually treated. It also allows for a mechanism for investigation and collaboration amongst pharma, cancer centers, ourselves as technology developers, and the other members of this ecosystem, so that these discoveries can move very, very quickly from just a finding through to something that can be brought to bear on many patients across the world. And now switching to our third topic, this is molecular monitoring, and this is basically using deep sequencing to detect and monitor cancer. What we're going to talk about today is the deep sequencing of cell-free DNA. Many of you may be familiar with the huge change in trisomy testing, bringing NIPT on board, looking for fetal DNA within the maternal DNA bloodstream. Very similar concept to what we can do in cancer as well, sequencing the fragments of DNA that are floating around in a patient's bloodstream, and being able to use that information to detect indicative somatic variation. Pardon me. So one of the important questions is, can we detect cancer earlier? And so we hinted at the beginning of this program that despite population-level screening programs, we're still, as a community, detecting many, many cancers at late stage, which limits the treatment options available for those particular individuals. And this is a statistic from a recent report from the CDC that almost half of the colorectal cancer and cervical cancers, and a third of the breast cancer cases in the United States are diagnosed at late stages. Late stages meaning that cancer has metastasized and spread to other parts of the body. And why? Well, there's a number of reasons for this. One is lack of compliance, for example, or lack of access based on where patients live or the health care that they have available. Lack of a screening test. Many cancers don't have a screening test, or certainly not a population level screening program in place. And then poor sensitivity of some of these tests as well. So what we're proposing here, and what others are proposing, is can we use the power of deep sequencing to develop a screening test that was non-invasive, for example, didn't involve a colonoscopy, was low cost, easily performed, and available for a broader range of different cancer types. The idea here would be to use blood, which is a readily available tissue and highly informative as a source for biomarkers that may be a solution to this problem. So there's a number of different groups that are really blazing the trail in this particular area of inquiry. And I'll highlight a couple of them in this talk. One of them is a group out of Johns Hopkins, and this is a paper by Bettigauda earlier this year that used whole genome sequencing and whole exome sequencing to identify driver mutations from tumor samples in about 600 different patients. They then used this information to inform the development of targeted sequencing panels to identify those same driver mutations identified from the tumor samples in the plasma of those individuals, from the plasma DNA of those individuals. And they were able to show that the presence of the tumor DNA using this approach they were able to detect somatic mutations from the tumor sample DNA at early stage in the plasma. And this is a significant finding because there was always the question of, well, perhaps you can only detect tumor DNA in the plasma of patients at a very late stage. How informative is that if the patient's already very sick? So this particular study, and you can see in the figure on the left-hand side, shows that in localized disease, which we're equating to earlier stage disease here, you can still detect the presence of tumor somatic mutations in the DNA at a high frequency. And so this would suggest the extensibility of these approaches to earlier stage patients. It's a very important proof of principle study to suggest the true promise of this approach going forward, and certainly this will be followed on by larger studies that look deeper and earlier into the plasma of various patients. Another study that also came out of the Johns Hopkins group, and this is an older one from a few years ago, but I think a really nice one that highlights what is possible and sort of the types of signals that one can identify from plasma DNA. In this one, Leary et al. Used whole genome sequencing of plasma samples to develop a plasma aneuploidy score. And they then applied this to a series of normal individuals, as well as a series of late-stage metastatic breast and colorectal cancer patients. Interestingly, the range of tumor fraction within the blood across these very late-stage patients was highly variable, ranging from about 1.4% to about 50% of the floating plasma DNA, DNA in those samples. But I think the graphic from this paper is extremely informative and highlights the power of this approach. You can see in the black bars that are on the left-hand side of the graph, those are the signals of this quote-unquote aneuploidy score from the normal samples included in this study. And you can see that they are nearly flat across the board. This is in contrast to the metastatic patients, the colorectal cancer patients and the breast cancer patients, which across the board have signals off the charts, if you will, here, but a clear separation between what is normal in terms of aneuploidy in the bloodstream versus what is indicative of cancer in this particular study. A limited proof of principle a study only looking at 20 patients, but I think a really nice and clear signal of the power and the potential of this approach going forward. Another aspect, another application for deep sequencing of plasma DNA is monitoring cancer in real time. So in this study, researchers are investigating the sequencing of cell-free DNA from plasma for monitoring as well. This is Newman et al., published earlier this year in Nature Medicine, and they use targeted sequencing of plasma samples to monitor tumor burden during treatment. What's really interesting here is you can see how they compare the signal of somatic variation in the bloodstream, which is the red line in both of these graphs here, to alternative methods for monitoring the effectiveness of treatment. In this case, the blue triangles are referring to the volume as established by imaging methods. And so in the first panel, in panel A, you can see that the molecular signature from the plasma DNA drops to 0 and is correlated roughly with what they're seeing from the tumor volume data as well. Similarly in B, so you can see very close correlation between the amount of circulating tumor DNA in the blood and what the volumetric analysis of the tumor via imaging is delivering to the physician at the same time. So mutation information from sequencing of plasma DNA correlates well with tumor volume in this particular data set. What's perhaps more interesting is when they disagree. And so researchers are investigating whether or not we can use this information as an additional line of evidence in conjunction with imaging information. And sometimes they don't agree. So Newman et al. Looked at these other patients where in the first panel, in panel E, you can see that their molecular signature drops to 0 at the time point marked number 4, whereas the tumor volume stays relatively high. And so if all one had to go on was the tumor volume, you would perhaps think that this patient was not responding to the therapy. I saw a recent presentation by this author at ASHG recently, and in fact this particular patient continues on today and is disease-free more than a significant amount of time later. In panel F, the opposite phenomenon is occurring. The red line is the plasma-based circulating tumor DNA metric, where you can see it's almost unchanged in response to therapy, versus the tumor volume has decreased to 0. So again, if the practicing physician were only looking at the tumor volume as the measure of whether or not the therapy's been successful, they would think that they had complete remission in this particular patient. Unfortunately, this patient did not show remission and then had progressive disease and then eventually died from his cancer later on. And so in this particular instance, this suggests that the plasma measurement was a more sensitive measure of disease burden in this patient, and is important information potentially to keep available going forward. So another open question in the space is how many different cancer types do we think are releasing their DNA into the bloodstream, and so how widely applicable is plasma-based sequencing? And a study, again out of the Johns Hopkins group, that looked at many different tumor types, suggests that the presence of tumor DNA in the bloodstream is a widely consistent phenomenon across different cancer types, with the exception perhaps being of brain-based tumors. And the range in terms of tumor fraction within the plasma is highly variable, as shown on the bottom panel. But clearly across a wide range is amenable to next-generation sequencing approaches. They also showed some really interesting data in this paper from earlier this year of the potential to use plasma-based sequencing to monitor for drug resistance, emerging drug resistance. So in this figure from the paper, they have a series of patients, colorectal patients, that have been put on EGFR blockade treatment. And you can see pre-treatment, they have no mutations in their KRAS gene or PIK3CA genes. When we look at their mutational status in their bloodstream post-treatment, in fact, many of these patients have now shown evidence of canonical resistance pathways kicking up, mutations in KRAS, for example, or additional mutations in the EGFR gene that would render their current EGFR blockade therapy ineffective. And so this is, again, a small study, only a couple dozen patients, but suggests that potentially deep sequencing, serial deep sequencing of known resistance genes throughout the course of therapy can be an early indicator of emerging resistance and potentially be useful for switching therapies down the road. Now, there's a huge amount of clinical research and exploration that has to be done here to truly understand the potential. But these early proof of principle studies, I think, are really exciting in the suggestions that they make for the future of this field. So to wrap things up, I hope I've given you a whirlwind tour of some of the things that we're excited about and working on here at Illumina, and the areas that we hope to be developing technologies in going forward: germline variation, further exploration of tumor biology and tumor profiling, and then molecular monitoring, and using our technology, deep sequencing across all three of these sort of areas of exploration going forward. And with that, I'd like to thank you for your attention, and I'd be happy to take any questions. Okay. If there's no questions, then I will wrap things up, and I thank you so much for your participation and your attendance today, and hope to see you again. Bye.